Good morning, ladies and gentlemen, and welcome to the 4BASEBIO PLC investor presentation. Throughout this recorded presentation, investors will be in listen-only mode. Questions are encouraged. They can be submitted at any time via the Q&A tab that's just situated on the right-hand corner of your screen. Please just simply type in your questions and press send. The company may not be in a position to answer every question it receives during the meeting itself. However, the company can review all questions submitted today and will publish those responses where it's appropriate to do so on the Investor Meet Company platform. Before we begin, I would just like to submit the following poll, and if you could give that your kind attention, I'm sure the company would be most grateful. I would now like to hand you over to the executive management team from 4BASEBIO PLC. Heike, good morning, sir.
Thank you, Jake, and thank you everybody for joining us today for the usual annual company update. We're excited to have so many people join us, and there's quite a few positive messages to bring today. Before we start, we have the usual disclaimer, so I'll give you a couple of seconds to just read through that. And then we'll start with introducing ourselves. So you're very familiar with David and myself. We've done a few of these presentations before, but today I would like to introduce you to Dr. Amy Walker as well, who will be joining us in this presentation and provide some further detail on some of the commercial development and competitive differentiators of our technology. So Amy, if you can introduce yourself.
Yes, so my name is Amy Walker. I am the VP of Research and BD here at 4BASEBIO. My background is in molecular biology and non-viral delivery systems, and my core responsibilities at the company are the development of our two technology platforms and their commercial development.
Okay, so today I'll start off with a company overview, just giving you a bit of a scope of where the company is at. Most of you are probably are long-term holders, but for those of you who are new to 4BASEBIO, we'll just introduce the company a little bit. As mentioned, Amy will then talk a little bit more about the competitive differentiators and the commercial progression that we've made. And then finally, David will give you an overview of the financials, and we'll close with a summary and some outlooks on 2024. So first of all, what is 4BASEBIO and who are we? We're essentially focused on two platforms. We want to bring synthetic DNA to the market, to the cell and gene therapy market, but also focus on delivery of nucleic acid payloads and have some solutions around that too. As you all know, we originally incorporated and started in Germany as a German European stock corporations, but we've now been in the UK for quite a few years, and we've been AIM listed since 2021. As a company, we've progressed quite a bit over the last few years. We've now grown to over 100 FTEs. In terms of facilities, we now have in excess of 23,000 square feet of facilities, incorporating R&D, commercial and manufacturing capacity. And we've been building our 7 grade C clean rooms as well, which is part and parcel of our infrastructure in synthetic DNA. So this kind of slide gives you an overview of who we are and the platforms that we're building on, right? So as mentioned, synthetic DNA is kind of our main commercial driver at this point in time. And our objective there is to replace plasmid DNA as the incumbent nucleic acid source in the cell and gene therapy space. Really driving forward the benefits of our platform in terms of cost effectiveness, speed, flexibility, and safety of the products that we make. We feel we can really drive forward the application and the adoption of our products and become a leading force in the cell and gene therapy market. Hand in hand with that goes, of course, the delivery of these payloads, and a lot of our clients have a need for payloads, but they also have a need for delivery of these payloads into patients and in the right places into patients as well. So very early on in the company's development, we've invested in a nanoparticle system that will allow us to do that, and Amy will provide a bit more detail around some of the benefits and focal points of that platform. Now, if you look at the bottom end of the slide, you will see that as a company, we're very much an innovation-driven company. You can see this, if you look at our workforce, for instance, 80% of our staff hold scientific degrees, with well over 20 PhDs as well. So very much, a scientifically driven entity, and all that kind of scientific power leads to a lot of innovation and generation of new intellectual property. As a result, we now have over 20 patent families covering a broad range of our portfolio, be that enzymes, be that processes to make DNA or certain specific DNA molecules as well. So there's a lot of breadth in the IP that we've built up over the past few years. And of course, that's supplemented with a lot of know-how that we've been building up over the last few years as well. I think, we can see some recognition of the platform in the industry and in the market as well. We recently won the Best Technology of the Year award at the MediSciences Awards recently here in London in June this year. So it kind of shows that the industry is noticing our platform and recognizing the value in what we do. Of course, it's not just all about science. We have to think about commercialization and manufacturing capabilities as well. So we've invested quite heavily in building all of that up too. As mentioned in my first slide, we have the Grade C clean rooms up and running. We have the QA teams and the manufacturing teams ready to go. And alongside that as well, we've been investing in the commercial infrastructure. And that is, of course, then both the BD team, but also the project management teams who help us with client onboarding and managing our clients through their journey with the company and ensuring that we have happy clients that come back for repeat orders. So all of that kind of shows you that the company is maturing and is developing from a kind of a research and development company into a fully fledged commercial entity that is able to onboard clients, deliver product, and drive its revenue and profitability forward. So in terms of key milestones for 2023, so revenue has doubled. So I mean, it's from a fairly small base. So from my perspective, what's more important is actually that we had a good influx of fee-paying clients and fee-paying projects in 2023, and of course that will lead to repeat orders in 2024, and we want to continue building that funnel and driving new projects into the business as we build up our client base. As a result of that, we've sold our first high quality grade material. We also had the first order for GMP grade material, and all of that, of course, was done through expanded headcounts. And as mentioned in the first slide as well, IP is important to us, and we continue to file new patents protecting our intellectual property space, and that has continued in 2023 and will continue in 2024 as well. I think this slide is quite an interesting slide, and it kind of shows you the makeup of the company and how the company has evolved from an R&D entity into like a more mature commercial entity. And I think if you look on this slide here, you can see that over half of the kind of FTE count is now fully engaged in commercial activity. So we've got our client projects teams. That incorporates, of course, the BD and the PM teams, but also the MSAT teams and the MPD teams that help, onboarding the projects and delivering the first batches of material. And then, of course, our QA manufacturing team, which is focused on high quality grade and GMP grade manufacturing and all the quality aspects involved around that. Of course, we still do a lot of R&D as well, and you can see the application validation teams and the platform development teams constitute about a third of the size of the company. And there again, we focus on upstream R&D and downstream R&D, but also on application validation and opening up new markets for new DNA modalities that we plan to manufacture in due course. So maybe a little more on our competitive positioning and the market opportunity as a whole. So this slide here kind of shows you the market that we're feeding into. And I think, the cell and gene therapy market, of course, is the core market that we want to supply DNA into. It's a very rapidly growing market with a compound annual growth rate in excess of 20%. And for us, the most important part of this market is that there's a lot of opportunity in the earlier stages of development. So there's a lot of candidates that sit in the kind of development phases, be that like late preclinical, early preclinical, or early phase 1 clinical trials. And that's the market segment for us that we want to dive into and where we want to convert clients and then grow with them through their kind of clinical development journey. So a lot of opportunity. The also good part of this is that the market is fairly differentiated. So you can see at the top of the slide, there's quite a few buckets. So you have mRNA, gene editing, AAV and lentiviral, both viral vectors essentially, and DNA vaccine. So for us, it doesn't really matter so much which application becomes more dominant, because all of these application areas require DNA at some stage in the manufacturing. So for us, we can really be quite differentiated in terms of applications that we feed into and really drive value out of all of those buckets. Maybe also quite interesting to mention is the kind of mRNA vaccine bucket and mRNA as a whole. So you can see, whilst these other markets are driving quite a lot of growth, mRNA, because of COVID, has had a big surge, and of course now the COVID revenue is somewhat tailing off, but of course there's a lot of new opportunities coming through, and for us that's actually the more important part of that market, is those new opportunities, those new programs that are being developed. That's where the opportunity sits for 4BASEBIO to supply DNA into, because evidently we are not going to change the DNA source in the Pfizer vaccine from plasmid to synthetic DNA. So there's strong growth in the cell and gene therapy market, drives the growth in plasmid demand. And if you look at the market size for plasmid DNA by 2034, this is now anticipated to be around $15 billion. And we believe that synthetic DNA, and particularly 4BASEBIO DNA, can take a leading kind of position within that market and within that next surge of DNA demand. Simply because we still see the same problems with plasmid DNA in terms of the scalability, in terms of the safety aspects, and in terms of the lead times in particular that clients are waiting for their DNA, and that becomes quite a bottleneck in the development of these therapies. This slide here is kind of demonstrating the flexibility of our platform. Amy will talk in much more detail about what these different modalities do, but I think here again we wanted to highlight the different buckets and the opportunity within these different buckets as well. And you can see particularly with IVT mRNA and with viral vectors AAV and lenti, a lot of new clinical programs are being started, and a lot of opportunity for synthetic DNA to take a strong position within those opportunities. In the second column, you can also see, or the third column, I should say, you can also see a lot of high-profile companies. So, we're engaged with a lot of these clients already, and we hope to switch them over to synthetic DNA in the near term. And then finally, before I hand over to Amy, some competitive positioning in terms of the DNA market. So what you can see here on the left-hand side is kind of three buckets, right? So you have the incumbent plasmid DNA with the key players within that space, and Aldevron, particularly with as part of the Danaher group, having a leading position within that market segment. On the left-hand side, you can see the synthetic market play, and there's two kind of key market areas. One is kind of de novo synthesis. Those players do not really compete directly with us. They sit upstream from us, and they tend to focus on making small quantities of DNA, so sub 1 milligram amounts. So they're not really feeding into clinical trials, et cetera, but they sit upstream, and they are, potential template providers for us in making bulk scale DNA. So the amplification-based platforms really is the genuine competition for us. Really there you have PCR-based methods, which struggle somewhat with scalability and fidelity. So really we think the RCA-based platforms, such as our own platform and of course Touchlight's platform, are the real technologies that can deliver the quality and the quantity of DNA required for clinical use in cell and gene therapy. On the right-hand side, of course, you can then see how we differentiate ourselves against all of these areas, and you can see, we kind of tick all the boxes in terms of competitiveness, flexibility, and speed at which we can operate, fidelity and quality of the product. So we think we're very well differentiated and well positioned to be the leading platform within the synthetic DNA space and the DNA space as a whole. That said, I'll now hand over to Amy, who will talk a little bit more about competitive differentiators.
Thank you, Becky. So we wanted to briefly highlight some of 4BASEBIO's competitive differentiators as compared to the incumbent plasmid DNA. As Heike mentioned, plasmid DNA is the predominant DNA source feeding into CGT and vaccine applications, and this slide really aims to position the benefits of our synthetic DNA technology. So firstly, we will start with cost effectiveness. By nature, enzymatic processes have significantly lower and capital demands with reduced process volumes and waste streams associated. Moreover, 4BASEBIO is uniquely positioned against other synthetic providers as we actually manufacture the enzymes that are required for our synthetic DNA manufacture. And this further allows us to reduce costs and allows us to control the quality of such production. The next point to mention is the flexibility of our scale of manufacture, and we are able to precisely tailor the scale of the reaction suited to our clients' needs. So our GMP offering ranges from 50 milligrams right up to 10 grams, and the lower of which is ideally suited for personalized medicines such as personalized cancer vaccines, or really for those early phases of clinical trials where many of our clients at these early stages do not need multi-gram batches of DNA, which is the typical output for conventional plasmid manufacture. Next, I wanted to highlight the flexibility of our platform. So Heike already mentioned that we make 4 different flavors of synthetic DNA, and each of these DNA molecules has application-specific benefits for the downstream user. So to highlight that, we would, for example, not sell the same DNA product to an mRNA client as we would to a viral vector client. And really, the flexibility of our platform resonates with our end users and is a key differentiator against other synthetic players within this space. And we have a more streamlined approach to producing these kind of custom molecules, which allows our clients to realize both cost and performance benefits in the downstream application. Next is safety. In summary, we are a bacterial fermentation-free platform, and so we're free from all of those contaminants associated with conventional plasmid manufacture, not limited to endotoxins, to host proteins, or indeed, and critically, to antibiotic resistance genes. Next, and which is a real driver of client conversion and adoption, is actually the speed and efficiency at which we can manufacture synthetic DNA. So this is really in a matter of weeks as compared to the typical 6 to 9 months associated with plasmid approaches. And this allows our clients to realize benefits in 2 forms. So on the one hand, they are able to continue to progress their development journeys without locking into a GMP manufacturing slot. But on the other hand, it allows our client base to expedite their journey towards the clinic as compared to conventional plasmid manufacturing. And finally, to point out on this slide, our enzymatic approach is able to deal with very complex sequences that are typically challenging in conventional plasmid fermentation approaches. So this could include homopolymeric traps required for mRNA production, for example, or ITRs for viral vector production, which are inherently difficult to propagate in bacterial systems. So switching tacks a little bit and moving on to our second technology platform, which is Hermes. Hermes is, of course, our nanoparticle system for the delivery of nucleic acid payloads. And one of the USPs of this platform against conventional LNP delivery systems, such as those used in the Pfizer-BioNTech nanoparticle COVID vaccines, are actually the enhanced stability that we have at both fridge and room temperature, which really omits the need for cold chain requirements necessary for the wide-scale distribution of such vaccines. Recently, there has been a significant uptake in using the Hermes delivery system for vaccine applications, both in infectious diseases, but also in personalized cancer vaccines. So we've really felt that over the last kind of 6 to 12 months. I think another USP with our platform as compared to a conventional LNP is the potential to target these nanoparticles towards specific tissues or organs of interest. And this is unlike conventional LNPs, which typically exhibit a tropism towards the liver, when they are delivered via IV administration.
Sorry, I just want to interrupt for a minute. We had one of our guests complain about the sound. I'm not sure whether Jay can do something about that and increase the microphone sound.
Hi, Heike. I can confirm the sound is clear. If we could maybe move the laptop just slightly closer to Amy, that would enhance the sound quality.
Okay, we will do that.
Thank you.
Thank you. So, I think to summarize on this slide, the platform has really progressed significantly over the last 12 months. And this has really been driven by funding from the Bill and Melinda Gates Foundation to combine our 2 core technology platforms to develop a thermostable vaccine using our synthetic DNA technology and our Hermes nanoparticle delivery system. So funding from such a prestigious institution has provided further validation for the platform. I think lastly to point out here, the platform remains synergistic with our synthetic DNA offering. And we have a growing number of examples where clients approach us with initial interest in either the synthetic DNA platform or our Hermes delivery technology, and actually end up evaluating both platforms. Next slide, Mikey.
Yep.
So changing tact a little bit, onto an update in our commercial journey and our revenue visibility over the last maybe 12 to 18 months. So to start with, we thought it would be useful to position our go-to-market strategy and how we anticipate the evolution of our client relationship cycle as we progress as a company. So as we've mentioned several times, synthetic DNA, of course, is a novel DNA modality. And really the main objective in 2023 was to make our clients comfortable with the use of synthetic DNA. So historically, we have picked up clients fairly early in their development stages, where they typically would evaluate our synthetic DNA against conventional plasmid. So these were typically research use only materials, And this could come in the form of an off-the-shelf construct, which is used for mRNA production or viral vector production, for example. But more often than not, our clients really prefer that we encoded their custom sequences so they could evaluate them in their in-house processes. In these cases, clients typically ordered a low mass of DNA, maybe 1 to 10 milligrams at research grade, and would typically evaluate 1 to 5 sequences, depending on the application area. The PO values were modest with respect to these projects. I think, of course, the objective of such evaluations is to convince clients of the benefits of synthetic DNA and then remain sticky through their development as they progress towards late preclinical and ultimately clinical applications. This year, we achieved a significant regulatory milestone with our first client entering the clinic with an mRNA vaccine derived from one of our synthetic opDNA templates. And I will talk about that a little bit more in the next couple of slides. But we've also seen over the last 6 months a real shift in our customer entry point to later within their development stages, with clients reaching out for high quality or indeed GMP requests for material. Of course, the value of such POs is substantially higher than with research grade material, and our objective over the next 12 months is to onboard more of these clients with HQ and GMP requests. So moving on a little bit to our client base, if we start at the left-hand side, we're looking at a pie chart that divides our client base by application. And you can very quickly see how many mRNA clients we have. So this really makes up almost 60% of our client base. You can see very nicely that that segment of the pie is split between clients that are working on vaccine applications versus clients that are working on therapeutic applications. So this could include gene editing applications where people are using our synthetic DNA templates to manufacture mRNA for nucleases required for such approaches. The next biggest segment there is our viral vector clients, specifically with the focus of AAV. And we have a growing number of clients in this space as well, who typically have higher mass demands of high quality grade DNA. And then finally, the remaining 20% or so of our client base is comprised of DNA vaccines, which is a very important segment for us, then for gene editing repair templates, and then some other more obscure requests for our synthetic DNA. Moving into the middle section, we are looking at the client base divided by region. And perhaps unsurprisingly, the majority of our clients sit in the US, particularly in Boston, with the remaining 50% divided nicely between the EU and UK. We also have a growing client base within APAC regions, and this is typically generated by inbound requests for our synthetic DNA templates. And finally, on the right-hand side, we have divided our client base by client type. So we do have a large amount of biotechs who are typically quite quick to pick up on novel DNA technologies that can expedite their development programs. But what we've really seen over the last maybe 6 to 12 months is growing uptake within larger and inherently more conservative areas of the industry, such as pharma clients who are trying to understand where synthetic DNA fits into their growth and development. Finally, the last part of this pie is really made up by CDMOs and strategic who hope to position synthetic DNA to a wider client base. So over the next 2 slides, we wanted to highlight 2 recent customer case studies and the journey that they've made from evaluating research grade material right into the clinic in this case. So in the first instance, we'll talk a little bit about a biotech company. This is a US-based biotech working on mRNA vaccines for various different applications, but initially for infectious disease. And the kind of profile of the molecule we made was a critical starting material opDNA template for their mRNA vaccine. So you can see that the relationship with this client, it started back at the end of 22, where we reached out to the client and convinced them to evaluate our synthetic DNA offering in a range of their applications. And you can see that by January, we were able to ship them those initial constructs for evaluation. In parallel, the client was very pleased with how the product performed in their internal processes. And so we received a PO for high quality grade DNA in January that year, which we later delivered to the client in June. And this material really fed into all of their GLP tox studies and their IND enabling studies. And then you can jump to March of this year, where, of course, this client ended up dosing patients in a phase 1 clinical trial with the mRNA vaccine I spoke about previously. So again, this was a very significant regulatory milestone that we achieved. I think jumping down to the bottom of the slide, you can see how this tracks nicely in parallel with their GMP requests. So this particular client wants to facilitate an FDA-based IND enabling study to facilitate a phase 1 clinical trial in the United States. And to that end, we received a successful on-site client audit earlier this year in February with the intention to manufacture a GMP batch later this year ahead of their phase 1 trial in the US. So next is at the other end of the spectrum. This is actually a pharma company. This is a global pharma company that was interested in using synthetic DNA for their seasonal vaccines and pandemic responses, where significantly reduced timelines is critical in being able to develop and deliver these vaccines. So this was actually an inbound customer request. We were approached by this pharma company who was interested in evaluating synthetic DNA in their seasonal flu programs, and we understood that we were being benchmarked against some other competitors within this space. So the first objective at the end of last year was to deliver a research-grade batch of opDNA, which we were able to turn around from receipt of sequence to the product shipped in less than 5 weeks, which was one of the metrics on which the client was evaluating us to determine success. To cut a long story short, this DNA molecule performed very well in their hands, and we received a quote for, we sent the client a quote for 2 high quality and 2 GMP batches earlier this year, which we're working on. Most recently, we had a successful audit from the client next month who came to visit our facilities, and we're happy with how they looked. So, I think to summarize our commercial progress, we have seen significant momentum in client engagement over the last 18 months. And this is really driven by both new contract wins alongside repeat orders from clients progressing towards the clinic. So you can see on this slide that the revenue generated from projects in 2023 was primarily driven by research-grade projects and those early development evaluations that I mentioned, alongside a handful of HQ clients. We anticipate that over the course of the next few years, we will have a significant evolution in the makeup of our revenue, with more substantial revenue share coming from these HQ and GMP contracts as clients progress through the phases of their clinical trials. Our expectation is that a 12 to 14 million revenue target is required until break even, and we estimate that this will take around 10 to 15 HQ or GMP projects per annum, allowing us to reach this next value inflection point in the near term. I will hand over to David for the financial overview.
Thank you, Amy. Good morning, everyone. I guess before we start, just very briefly, I just want to touch on some of the points I think Heike and Amy have made, and in terms of the progress we've made over the last year. Really, those operational, clinical, and customer-focused developments don't necessarily reflect themselves in the income statement and the cash flow that I'm about to share with you, but it really sets us up extremely well moving forward into 24, 25 and beyond. So moving on to the financials, really the income statement for 23 was very much as expected. We obviously have a budgeting cycle. We set our budgets and plans and work on that basis. And the result overall is, as I said, very much consistent with that plan. The main feature, which I think Heike has alluded to, is where we are in terms of our revenue. And while the absolute revenue number has doubled, clearly it's from a very modest base. But much more significantly, as Heike's highlighted, it's a number of individual projects that we've managed to undertake during 2023. And clearly, we expect to grow that strongly during the course of 24 and beyond. Overall, in terms of cost base, there's been a step change from 2022, but I think we're gradually reaching a steady state. So while we expect there to be additional heads and additional investments in the cost base over the course of 24, really, we are now reaching a close to a mature, steady state position. Inevitably, you don't have within 23 a full year of cost reflecting that growth, but during the course of 2024, that we will reach that point. We had a headcount during the course of 2023 of 86. We expect to grow that over the course of this year to around 110. And that really will hold us in good stead going forward. In terms of the cash flows, I guess the cash flows are very much as expected. They obviously reflect the operational expansion of the business. And in addition to that, We've had to draw on the to invest facility, I think, as we indicated earlier in the year, and that has really funded the development and the growth during the course of 2023, and we expect to draw on that and use that during the course of 2024 as well. Can you move on? In terms of the balance sheet, I'm not sure how much there really is to say about this. The assets clearly reflect further investment, so you can see, particularly within our property, plants and equipment and our ongoing intangible assets capitalization. And we continue to invest both within the property, so the expansion of the facilities supporting our clean rooms, and also platform development across the board. Closing cash for the period was 3 million. Clearly, in order to fund operations during 24, as I've just mentioned, we will draw on the to invest facility to support that cash position. In terms of liabilities, the liability position really reflects the use and the utilization of the to invest facility. So you can see within our non-current liabilities, the financial liabilities have stepped up. This represents the drawdown of amounts from to invest. Those facilities are available to us until 2028, where repayment is due in October 2028. So clearly it's not an immediate issue. And really, as consistent with 2023, we will in 2024 use those facilities, as I mentioned. Finally, looking to the financial overview and outlook for 2024. We really do expect strong revenue growth during the course of the year. The nature of some of the GMP and HQ projects means that this revenue growth can be quite uncertain, both in terms of the timing of customer projects, but also it can be relatively lumpy in terms of overall revenue and when it hits. Relative to that, we can see strong growth in the order book. The question is really when that hits during the course of the year. But overall, strong revenue growth expected. We will continue to invest in our operating expenditure. As I mentioned, we're going to move from 86 heads to around 110. And that, as I say, will be a largely steady state position from which we can then continue to develop our revenue during the course of 2024, 2025 and beyond. And finally, in terms of the funding of that position, at risk of repeating myself again, we will draw on the to invest facility to enable the ongoing growth of the business.
Thank you, David. So yeah, then we're rapidly moving into the final section of the presentation. I mean, on this slide, I kind of wanted to take you through the journey that we've been through over the last few years and the kind of value inflection points that we've achieved, and also the inflection points towards adoption that we've achieved over time. So, of course, it started off with, kind of the scale up and the application validation. So our application validation teams have generated a lot of data around these different application areas that we want to feed synthetic DNA into. We've got really good data packages that is very convincing to clients and is a real big stepstone to getting conversions. And alongside that, of course, we've had to scale up our platform too. I mean, when we started off in 2020, we were making DNA at microgram scale. We might now make DNA in multigram batches. So you can see we've achieved, a million log, kind of a million times uplift in terms of scale. So that's pretty impressive. And the entire platform as well is, kind of dedicated and focused around single use, so that we don't have to worry about about cross-contamination and can really focus about quality, around quality of our DNA products. That said, now our focus has really turned towards our GMP readiness, and I think there's probably a bit of context that needs to be understood around GMP readiness as well. I mean, our clients that require different grades at different stages and for different applications within their own development program. So, I mean, typically, when clients go through GLP tox studies, so that's kind of late preclinical studies, they will require high quality grade batches from us, which are kind of manufactured in a similar way compared to the full GMP material, but doesn't have all the bells and the whistles. But in terms of GMP material material as well, clients need different things. Our mRNA clients and our viral vector clients need starting material. So essentially our DNA is converted in a manufacturing process from DNA to mRNA or from DNA to a viral vector, but our DNA product is not part of the final drug substance or drug product that our clients are making. So we're already in a position to make critical starting material. Of course, the next step for us is to think about drug substance, where our DNA product does become part of the final product that will be administered into a patient. And that's kind of the next step on our kind of regulatory journey, is getting that MHRA certification and that license for drug substance. Overall, I think from a commercial perspective, we can see good traction building, and we can kind of feel we're in a good position to start displacing some of the incumbent materials within the market, and then ultimately drive into commercial development as well, and getting products into patients on a regular basis. So in terms of objectives for 2024, we want to continue to build on the momentum that we see within the market and drive forward our revenues. We've got very good GMP visibility now. So, in 2023, we just started going into the market. We started getting some clients in. We're now seeing a demand for GMP building up. We see it's not necessarily an order book, but it's we refer to it as GMP visibility, clients that we know will need GMP grade material in the near term, and where we expect to be embedded within their programs. However, the usual issue with these things is that the timing and the cadence of these orders is client dependent. It depends on their progress, it depends on their funding. So as a result of that, we expect some unevenness in the kind of revenue recognition in the near term as well, as we get a more stable kind of order book building up. We want to advance our GMP certification, so our objective is for this year to get our MHRA IMP license. As I mentioned, we want to be able to make drug substance, and that's something that is a key objective for this year. And then, of course, we want to continue investing in our platforms as well, really focusing around process intensification, which will allow us to make bigger batches from our existing facilities, and also continue driving down our cost of goods as well. I mean, we're in a very good competitive cost of goods situation already as it is, but we feel there's scope to further improve this. And finally, we want to focus on new DNA modalities, be that single-stranded DNA or really advanced, enhanced DNA molecules that have performance benefits for our clients, and really opening up new market opportunities as a result of that, and position ourselves as the leading company within DNA and synthetic DNA in particular. So that said, in summary, I think it has become clear that we have a very unique company with some very strong technology platforms, both in synthetic DNA, but also in delivery of these nucleic acid payloads. And we have a very flexible approach to making DNA molecules, enabling us to really make these added value products and giving us a competitive position within the market space. And we've built up the infrastructure, both in terms of manufacturing DNA, but also in terms of commercializing, onboarding clients, and delivering high-quality products for our client base. So we feel we're in a very strong position to really drive forward revenues for the company in the near term, and bring the company to its break-even position, as Amy mentioned, that next big value inflection point that we now are trying to achieve. So with that said, I'll leave it here, and we'll switch over to the questions and answer session next.
Perfect. Heike, David, Amy, if I may just jump back in there. Thank you very much indeed for your presentation this morning. I will just bring up your camera there. Ladies and gentlemen, please do continue to submit your questions just by using the Q&A tab that's situated on the top right-hand corner of your screen. But just while the company take a few moments to review those questions that were submitted already, I would just like to remind you that a recording of this presentation, along with a copy of the slides and the published Q&A, can be accessed via your investor dashboard. Heike, Amy, David, as you can see there, we have already received a number of questions throughout your presentation this morning. And thank you to all of those on the call for taking the time to submit their questions. But guys, at this point, if I may just hand back to you just to read out those questions and give your responses where it's appropriate to do so, and then I'll pick up from you at the end. Thank you.
Thank you. So we'll start diving through the questions as they were filed. So we'll start off with the first question by Andy, who asked us whether the profitability of the GMP batches would increase over the profitability over the research grade batches. So yeah, I mean, GMP grade batches and high quality grade batches are in that same bucket. So our higher grades materials have really high margins. At that point, we've already had the kind of template generation costs embedded within the project, and we can drive really high margins of those batches. They're not really sequence dependent, so it really it doesn't really matter what type of sequence we make, whether it's a big molecule or a small molecule, whether it's GC rich or not, or whether it has ITRs or sequence complexity. And the process really is driving DNA to 1 gram a liter at this point in time, and we can achieve that irrespective of the sequence. Amy, is there anything you want to add to that?
Yeah, I mean, I think that's a very valid point, the sequence independency of the platform itself. So like Aki already mentioned, we can deal with these complex sequences, but we also don't have any size limitations. So we can make molecules as small as 150 base pairs, right up to 20,000 base pairs. And this really opens up significant market opportunity for us in, for example, applications where they need much larger DNA constructs, such as self-amplifying mRNAs. So that's a real driver for conversion.
Yeah, I think maybe one extra point to this as well is that, because we make our own enzymes, we control, of course, the quality of those enzymes, but also the cost of the enzymes is important. And for us, our enzymes, which is one of the key ingredients in making the DNA, is not a major cost driver for us anymore. We have other cost drivers that are more dominant. So we're in a pretty good position to really scale rapidly and become very cost effective and drive high margins at scale. So,
shall I pick on?
Yeah. So the next question, David, it was whether.
I'll read it out. So Jonathan's asked, the company's previously indicated it would be willing to raise equity to reduce dependence on Mr. Zurs at the £7 to £8 share level. What's the latest on this? I guess there are three aspects to that. In terms of raising funding, clearly we've always been open to doing that and been mindful of when it's appropriate. This isn't really about a question of reliance on Mr. Zurs. The Deutsche Balaton Group have always been very supportive of us and can continue to be so. So we will consider our options as and when it's appropriate. Relative to the £7 and £8 mark, I guess really the thing is, time moves on, the company continues to develop, and we become more ambitious in terms of the valuation of the company and what we would consider to be an appropriate price to raise equity. Having said all that, there are no plans to raise equity, but it is something we keep under review from time to time.
Thanks, David. So, moving on to the next question, which is also by Jonathan, about the timing around the GMP certification. So, as I mentioned in my last slide, the objective is to get the IMP license. So that's the investigational medicinal product license for drug substance. The timing of this is not entirely in our hands, right? So we're somewhat dependent on the MHRA as well, in terms of the timing around their audit and how fast they can progress an application. So, we're hopeful that we can get this achieved within 2024. But like I said, we're somewhat dependent on the MHRA and the progress they can make on this application as well.
Yeah, maybe we could add to that point that certification is critically important for our DNA vaccine clients who will actually be delivering the DNA that we produce as a drug substance. But for the majority of our clients that are using the DNA molecules to manufacture viral vectors or for mRNA, that is of less importance, as you can see with our progression into the clinics in these application areas.
Yeah, exactly. Thank you, Amy. So moving on to the next question by Andy, asking us whether the platform is generally sequence independent and whether we've tested this on longer base repeats and GC-rich regions, typically like the ones found in eukaryotic promoter regions. I'll give that one to Amy.
Thank you. Yeah, so I think back to our enzyme point, the enzymes that we manufacture are proprietary, but they've been highly engineered for both their processivity and their fidelity. So we have lots and lots of different examples of their ability to deal with these very complex sequences, including the GC-rich sequences that you mentioned. So GC content is really not a driver in the fidelity or the accessibility of our enzymes. And then, of course, there are the homopolymeric tracts, which are typical in mRNA constructs, for example. Again, we have no problems with these sequences, and we can encode into our DNA templates tracts upwards to 120 polyAs. So this is not significant for us. And indeed, we often see clients come to us with sequences that they just cannot propagate in conventional plasmid systems, so they move to a synthetic approach.
Yeah, thank you, Amy. So the next question was more about where we can capture revenue and whether it's an opportunity for us to capture revenue in phase 1, phase 2, or phase 3 clinical trials. So I think there, I think we have to be realistic. Like, we're still at the kind of early adoption phase of synthetic DNA. So of course, it's much easier to do this in the kind of late preclinical, early clinical stage, or early preclinical stages. That said, I think as we build up more momentum and we get the kind of regulatory validation, we expect more mainstream usage of synthetic DNA. And I think at that point, given the benefits of synthetic DNA in terms of lead time, cost, and performance, I think there is opportunity to start switching people over in phase 1 or maybe phase 2 clinical trials. I mean, the further up the development kind of track a given project has gone, the harder it becomes to convince clients to switch over. But I do think in time we will be able to see entry points of in later clinical stages as well. But I think at this point in time, we really focus on that kind of late preclinical stage, so that we can really follow our clients through their entire clinical development journey. Okay, so the next question is going to be another one for Amy. I think it's a question from Andy on targeted delivery of nucleic acids and whether that's a key challenge today, and if so, whether Hermes can deliver this kind of targeted delivery.
Thank you. So that's quite a difficult question. So I think conventional lipid nanoparticle systems, as I mentioned, have this inherent tropism towards the liver. And that's why you can see all of these new biotech companies that are focusing their gene editing technologies on liver diseases with really, really good progress. I think that there has also been significant development within the space as people try and detarget the liver and move to more complex organs within the system that are a little bit more difficult to deliver nucleic acid payloads to. And these particular clients may be using very complex manufacturing processes, for example, conjugating an antibody to the surface of the nanoparticle or an antibody fragment. I think the real differentiator with Hermes, which I did not mention in the slides, is actually the kind of ease of manufacture. So we are able to incorporate this targeting moiety into the lipid nanoparticle via microfluidic manufacture of the particles, which really helps us to be a very cost-effective and scalable process. I think to your question on what sort of organs that we target, or we can't disclose all of those, but we do have some programs running in cell therapy approaches, ex vivo and in vivo. We have a growing number of clients trying to target the kidney and the heart.
Yeah.
And I'll leave it there.
Thanks. I would echo the words from Amy, but I think what we've also seen is a big kind of shift in demand of the nanoparticle system in the kind of vaccine space. I mean, originally we didn't anticipate that we would be competing within that space given the success of LNPs during the COVID pandemic, but we do see that there's a big pull for the next generation of LNPs within the vaccine space as well, which particularly kind of deals with the issue around the cold chain and the stability of the particles. So for us, that's been an unexpected opportunity, but it's looking actually very promising as an opportunity. Okay, so moving on to the next question. Jonathan asked us whether the level of indebtedness impact on our ability to sell to pharma and biotech. David, do you want to deal with that?
Yeah, I have to say it's never come up as a feature. Obviously, we have supplier onboarding processes we need to go through. I think the most colourful one so far is being asked to confirm that we're not a criminal organisation, and if we are, we're in big trouble. But really, we fill in these forms fairly regularly, and it hasn't been a feature.
Thanks, David. So next question, again from Jonathan, is whether we will need to submit a drug master file to the FDA in order for customers to be able to file INDs, and what's the timing on this? So that is not a requirement. So whilst we had that on our kind of wish list of things to do, we eventually actually focused our energies more on getting the IMP license and the MHRA certification, because we think that has much higher added value than having the DMF filed at the FDA, particularly for our clients, our pharma clients, and our higher level biotech clients. The IMP license is of higher value than the DMF. So it's something that we will do in time, but we focus on official certifications at this point in time. So next question was from Freddie. So how much of the opportunity pipeline is contingent upon achieving full GMP accreditation, and are you hoping for that to be achieved this current year? So the bottom end of the question, yes, we're hoping for this to be achieved this year. And again, in terms of certification, I want to make it clear we're going for an IMP license for drug substance. There's other things beyond that you can go for. For right now, for our client base, that is what we feel we need to open up some of that kind of DNA vaccine space and gene editing space. But that said, if we look at our GMP visibility list, a lot of our client base is within that mRNA space where you need a critical starting material. So for those clients, that IMP license isn't essential. So we can already sell within mRNA applications, within viral vector applications without that license. So as Amy's mentioned, a lot of our kind of existing client base and opportunity list, isn't needing that IMP license today. And then finally, another question from Jonathan, asking whether, well, actually, Emily, you can read the question. It's addressed to you.
Yes, thank you, Jonathan. So I was quoted in an article earlier this year that 4BASEBIO anticipates the entry of several additional clients towards phase 1 clinical trials. And can we expect further announcement this year along the lines of the Tier 1 pharma announcement? I think high level, yes. We anticipate a number of our clients to make an announcement as such. I think it's important to mention that the timing and cadence of this remains in our clients' hands. So whether that would happen in H2 of this year or maybe early next year remains to be seen. But we have several clients progressing nicely towards the clinic, and probably worth mentioning that these are in a range of different geographical jurisdictions, so under FDA, EMA, or indeed the MHRA. So we feel very positive about that.
Yeah, I mean, the only context I would set to this as well is that we're probably unlikely to be able to name the clients, because a lot of those clients tend to prefer confidentiality around that. But we're expecting to be able to make announcements similar to the T1 pharma announcements in due course. So I believe that was the last question. So back over to Jake.
Yeah, absolutely. And David and Amy as well, thank you very much indeed for being so generous with your time and addressing all of those questions that came in from investors this morning. And of course, if there are any further questions that do come through, we'll make these available to you immediately after the presentation has ended, just for you to review, to then add any additional responses, of course, where it's appropriate to do so. And we'll publish all those responses out on the platform. But Heike, perhaps before really just looking to redirect those on the call to provide you with their feedback, which I know is particularly important to yourself and the company, if I could please just ask you for a few closing comments to wrap up with, that'd be great.
Yeah, no, I'd like to thank everybody for joining the session today, and I hope you found it all very informative. I mean, the questions were very interactive, and we really appreciate you guys taking the time out to come and listen to us and ask us the questions. So we look forward to updating in due course, and we believe the company is in a great position to deliver, to continue delivering shareholder value as well. So we look forward to the next update. Thank you very much.
Perfect, Heike, that's great. And thank you once again for updating investors this morning. Could I please ask investors not to close this session, as you'll now be automatically redirected for the opportunity to provide your feedback in order that the management team can really better understand your views and expectations. This will only take a few moments to complete, but I'm sure will be greatly valued by the company. On behalf of the management team of 4BASEBIO PLC, we would like to thank you for attending today's presentation. That now concludes today's session, so good afternoon to you all.